So we are going to start today on the function of the major histocompatibility complex molecules. And since we have done the structural parts, we understood the function, we understood the structure of both class 1 and class 2 molecules. And to keep it in mind that when we enter the processing pathways, remember that class 1 molecule is ubiquitous in nature. It is present on the surface, it can be presented on the surface of all cells of the body, whilst MHC class 2 is present on the surface of antigen presenting cells. So this is one thing to be kept in mind. When we look into the two pathways which are operative with respect to MHC class 1 and class 2, the two pathways can be broadly divided into the exogenous pathway. In the exogenous pathway, as you can see, extracellular bacteria, parasite, toxins, they are ingested by the antigen presenting cells. Within the cytoplasm of the antigen presenting cells, the ingested antigens are degraded. Since these degraded antigens or polypeptides will be loaded on MHC class 2, remember the size of these polypeptides will be 13 to 18 amino acid long. And these 13 to 18 amino acid long polypeptides will be uploaded on the peptide binding groove of MHC class 2 molecule, which is formed by the alpha 1 and the beta 1 domains. And once this peptide is uploaded or loaded on the peptide binding groove of MHC class 2, the question is, to whom does these MHC class 2 present this peptide? Why is it displaying this peptide? So this Y is, it displays this foreign antigens to this category of T lymphocytes, which we had studied when we did our introductory class of the immune system. To recapitulate, I had said that the T lymphocyte population can be broadly classified into CD positive helper cells who have the CD4 as the co-receptor, and the CD8 positive T cytotoxic cells who have the CD8 as the co-receptor. So the CD4 lymphocytes recognize the antigen, and depending on the nature of the antigen, further activation on the T helper cells are seen. So that is how what happens whenever a foreign pathogen or foreign bacteria is engulfed by an antigen presenting cell, digested, degraded, peptide fragments are obtained, and these peptide fragments are loaded on class 2 groove and are presented to CD4 positive T helper cells. When we look on into the other pathway, you can see intracellular bacteria, virus, tumors. So something that is occurring in the cytoplasm. So these are intracellular. So it can be a self-protein getting abnormal. It can be a virus injecting its RNA or DNA. And as per the instruction of this viral RNA or DNA, the host cell machinery is now producing viral proteins. So these proteins are also foreign. So the moment this kind of proteins are present in the cytoplasm of that particular cell, what happens, these proteins are identified as foreign, and you can see these foreign proteins are degraded to a length of 8 to 10 amino acids. And once these foreign proteins are degraded to a length of 8 to 10 amino acids, they are then uploaded into the peptide binding groove of MHC class 1, which is formed by, if you remember, the alpha 1 and the alpha 2 domain. And these MHC class 1 molecules now present their peptides to the CD8 positive T cytotoxic cell. So this is a phenomenon known as MHC restriction. That means class 2 will always present antigens to CD4 positive cells. So class 2 is restricted to activate CD4 positive T helper cells, and class 1 is restricted to activate CD8 positive T cytotoxic cells. This is a very simplified diagram of what actually happens. We'll be going into the detail. So, but just summarizing what is going to happen, a virus enters. It might inject a viral DNA or RNA. So we are getting a viral mRNA. It is using the host machinery to produce proteins. So we are having the viral proteins. These viral proteins then enter the Golgi complex. Very sorry. They enter the endoplasmic reticulum. And within the endoplasmic reticulum, the newly formed MHC class 1 molecules are present. Within the endoplasmic reticulum, the peptide fragments are uploaded in the peptide binding groove of MHC class 1. And then you can see the MHC class 1 molecules are pinched out, moving through the Golgi complex and ultimately being displayed to the surface of the cell. So this is also known as cytosolic pathway or endogenous pathway. The other one is that, as you can see, a foreign pathogen has been engulfed, an endosome has been formed. Within this endosome, the foreign pathogen is digested, peptide fragments are generated. Meanwhile, MHC class 2 has been newly synthesized in the endoplasmic reticulum. They come out as a vesicle from the ER and move towards this endosome, and they fuse with the endosome. So when this fusion occurs, the foreign peptide fragments are then uploaded in the MHC class 2 molecule peptide binding groove, and the class 2 molecules then display these peptides to the surface. This pathway is known as we saw, exogenous pathway or the endocytic pathway. We will start our class today with the cytosolic or the endogenous pathway. So we did study cell biology in SEM 2. Yeah, SEM 2, and that time I think so during. Okay, so we studied this proteasome while discussing, as long as I remember, cell cycle. I talked about this ubiquitin and proteasome, so I'll not get into the details of all those. But for now, whenever there is an aberrant protein, a protein that is defective, is present in the cytoplasm, what happens? These ubiquitin molecules, they act as tags for aberrant proteins. How does the cellular machinery, how will the cellular machinery identify that which protein is correct and which protein is aberrant? So, to make the cellular machinery understand that this is a correct protein and this is a defective protein, the defective proteins are tagged with monomers of ubiquitin to form a polyubiquitin chain. So the proteins which are to be degraded, the proteins which are foreign, the proteins which are defective are tagged with these ubiquitin monomers. Once these proteins are tagged with these ubiquitin monomers, now these proteins are nothing but long polypeptide chains. So they are defective proteins present in the cytoplasm. They have been tagged with the ubiquitin complex. The question next arises is that I need to break these polypeptides into fragments of size, if you remember, 8 to 10 amino acids. I cannot have this defective polypeptides in this manner because I need to break them. So the defective proteins are tagged with the ubiquitin monomers, and the tagged defective proteins then enter this proteasome complex. As you can see, this proteasome complex where the defective proteins are degraded into smaller fragments. The proteasome complex, as you can see, has a 19S cap on two sides. It looks like a barrel. It is a barrel-like protein with size of around 150 angstrom to 115 angstrom in diameter. It consists of a 20S core. So this is 20S. And we have the 219S cap on the two sides, which acts as the regulatory protein. The core protein, as you can see, are stacked in 4 rings. Of these 4 rings, once more. Of these 4 rings, these 2 central rings are the most important ring, because as the protein enters into the center, centrally, it is this area which is the active site. It brings about the proteolytic cleavage of the target protein, and there is a threonine present in that active site. So the moment the ubiquitin tagged proteins enters this proteasome complex, as you can see, they come out as fragmented peptides. Now, there is a concept of immunoproteasome. You can see there are 2 categories of proteasome being shown here. One is the constitutive proteasome and one is the immunoproteasome. What's the difference? In normal cells, we usually have the constitutive proteasome. Okay, that is, they are already present, and if there is a ubiquitin tag protein, the protein enters this ubiquitin barrel, it is digested and fragments are produced. But in activated antigen presenting cells, where the cells are actively digesting, the cells have been activated by a microbe challenge. The immune system is already active. So in this activated antigen presenting cells, the constitutive proteasome, you look at the 3 subunits, beta 1, beta 2, and beta 5. These 3 subunits are replaced by another category of subunits, as you can see, beta 1I, beta 2I, beta 5I. This immunoproteasome has an increased efficiency for proteolytic cleavage of proteins, resulting in peptides that can be assembled with MHC class 1. Moreover, an important cytokine plays a role in formation of immunoproteasome. This cytokine is IFN gamma. In presence of IFN gamma, it has been seen that the beta 1, beta 2, and beta 5 subunits of a constitutive proteasome are replaced by immunoproteasome. So that was about the proteasome. So the moment the protein is tagged with a ubiquitin molecule, the protein enters the proteasome complex, and you can see the protein has been degraded into peptide fragments. So now let's move to the next part. So this is another picture showing you the immunoproteasome and the constitutive proteasome. And these are the different kinds of fragments that can be generated, both by immunoproteasome and constitutive proteasome. Now, as you can see, the protein is present. The protein was tagged by ubiquitin monomers, and this polyubiquitinated protein enters the proteasome complex or the immunoproteasome, and these proteins are degraded, right? So once these peptide fragments are generated, around 8 to 10 amino acids, these peptide fragments then starts moving to the endoplasmic reticulum. Just to keep in mind that when it is going to enter the endoplasmic reticulum, there are 2 transporter molecules present on the surface of the endoplasmic reticulum, which in an ATP dependent manner allows these peptide fragments from the cytoplasm to pass through it. One more point to be kept in mind here is that the peptide fragments that the TAP molecules can recognize are of 8 to 16 amino acids. Even though it can recognize fragments of 8 to 16 amino acids, keep it in mind that the optimal peptide length that can bind to MHC class 1 molecule is 9 amino acid. So as I was saying, this TAP or the transporter associated with antigen processing is a membrane spanning heterodimer consisting of TAP1 and TAP2. And this TAP1 and TAP2 has affinities for peptides containing 8 to 16 amino acids. These peptides then enter the endoplasmic reticulum. Within the endoplasmic reticulum, if longer peptides, as I said, the length of the peptide to bind to class 1 is 8 to 10 amino acids. But TAP has affinity for a length of 8 to 16 amino acids. So there is an endoplasmic reticulum aminopeptidase which can trim off the longer peptides to the desired length. So if you have longer peptides, those peptides are cleaved to give a desired length peptide, that is 8 to 16 amino acids. That is 9 amino acids. So once the peptides have entered the TAP, now comes the next part. So as you can see, the peptide fragments have entered the endoplasmic reticulum through the transporter associated with antigen processing. Meanwhile, the nascent MHC class 1 chain has been synthesized and is now present on the lumen of the rough endoplasmic reticulum. Remember, this thing is now happening here. The peptides have entered the endoplasmic reticulum lumen. So in the lumen, meanwhile, MHC class 1 alpha chain has been introduced, and the assembly process of the alpha and the beta chain involves quite a number of steps. As you can see, the first molecular chaperone. Now, these are known as molecular chaperone. The word chaperone means molecular bodyguards. These bodyguards helps the proper association of the MHC class 1 and the beta-2 microglobulin molecules. So the first molecular chaperone is Kynexin, which is a resident member protein of the endoplasmic reticulum. And ER57 is again a protein with enzymatic activity that associates, as you can see, with the free class 1 alpha chain and promotes its folding. So ERP57 helps in folding of the MHC class 1 alpha chain, and Kynexin acts as this bodyguard, and this is the complex thus formed. Next, what happens, as you can see in the picture, the beta-2 microglobulin is now entering the scenario. When the beta-2 microglobulin binds to the alpha chain, just look here, when the beta-2 microglobulin is binding to the alpha chain, you can see that Kynexin has opened up. It has been removed. So when the beta-2 microglobulin binds to the alpha chain, Kynexin is released and class 1 molecule associates with another molecular chaperone, calreticulin and tapacin. As the picture goes, so Kynexin opens up, tapacin and calreticulin, these are two other molecular chaperones, then binds to this complex. Tapacin, the word tapacin is an abbreviated form for TAP associated protein. What is the actual function of tapacin? The tapacin brings the TAP transporter. If you remember, this was the TAP transporter present on the surface of the endoplasmic reticulum. So what does tapacin does? Tapacin brings the TAP transporter into the proximity of the class 1 molecule and allows it to acquire one antigenic peptide. If you remember, we talked about the ERAP. It's aminopeptidase. It is the aminopeptidase of endoplasmic reticulum present within the endoplasmic reticulum. So if longer fragments of, as I said, TAP has affinity for 8 to 16 amino acid long polypeptides. So if longer polypeptides have entered the ER, they are trimmed by the endoplasmic reticulum ER aminopeptidase. So as you can see, this was the protein tagged with ubiquitin, peptides generated, longer peptides entering through TAP. The longer peptides are fragmented to desired line by the aminopeptidase present in the ER. Meanwhile, we have the assembled MHC class 1 complex. The tapacin has brought the MHC class 1 complex to the TAP transporter. The MHC class 1 molecule has brought the tapacin, the tapacin of the MHC class 1 molecule has brought it closer to the TAP transporter. And now, as you can see, the desired peptide of 8 to 10 amino acid is being uploaded into the groove of MHC class 1. Once the molecule has been uploaded into the groove of MHC class 1, all the bodyguards open up, as you can see. So the TAP protein promotes the peptide capture by the class 1 molecules. And once the peptide has been loaded, as a consequence, you can see this MHC class 1 molecule with the loaded peptide can now exit the endoplasmic reticulum. So as you can see, so the MHC class 1 has been loaded by the peptide by the process we just now saw, and gradually it moves out through the Golgi in a vesicle to finally be expressed on the surface of the cell in which this antigen processing and degradation took place. So that was about MHC class 1 molecule. Now we move on to the other pathway which I was talking about, the endocytic pathway or exogenous pathway. So when we are talking about the endocytic pathway, the exogenous pathway, what happens? If you remember, again I'm repeating, keep this in mind, these pathways operative only in antigen presenting cells. So in nature, in our body, all antigen presenting cells are capable of engulfing foreign pathogens. They have this property. Three classical antigen presenting cells are the macrophages, the B lymphocytes, and the dendritic cells, of which we had heard when we did the immune system cells and organs. So these are the classical antigen presenting cells of our body. These cells can engulf the foreign pathogen or the antigen. As you can see in this picture, what it has been shown, that the antibody present on the surface of a B lymphocyte has trapped an antigen. And the antibody and the antigen has been enclosed into a clathrin coated vesicle. And within this vesicle, you can see the antibody is being stripped off from the antigen. The antibody is recycled to the surface, and the antigen is now present in a small compartment, which is now endosome. In this pathway, one thing that happens is that the antigens move from one compartment to another, and as they move from one compartment to another, they move from a lesser acidic compartment to a higher acidic compartment. So as they move from one compartment to another, the pH of the compartment, initially which might be of 6 to 6.5, decreases to 4.5 to 5. So these are endosomal compartments. Finally, these endosomal compartments fuse with lysosome to form the endolysosome. Within the endolysosome, these protein molecules or the polypeptides are digested, proteolytically digested to a desired length of, if you remember, 13 to 18 amino acids. So we have these 13 to amino acid long fragments. Meanwhile, within the endoplasmic reticulum, the MHC class 2 molecules have been produced, synthesized. The MHC class 2 molecules come out. I'll be discussing the MHC class 2 molecule details just now. And they fuse with these endolysosomal compartments, where the fragments and these compartments are known as these MHC class 2 compartments are known as MHC class 2 containing compartments. So this fusion occurs and the peptides are loaded on the peptide binding groove of MHC class 2. Let's look into this picture. So as you can see, if we look into this endocytic pathway, this particular picture, as you can see, that the class 2 molecules are being synthesized and introduced into the endoplasmic reticulum. In the endoplasmic reticulum, there is another molecule which is known as the invariant chain. So when the class 2 molecule moves out from the endoplasmic reticulum, the invariant chain positions itself in such a manner that it occupies the peptide binding groove of the MHC class 2 molecule, so that by mistake another peptide fragment is not introduced in its peptide binding groove when it is within the ER. So the class 2 molecules are synthesized and the invariant chain is synthesized, and the class 2 molecules with the invariant chain comes out as a vesicle. Meanwhile, as we just now saw, the pathogen has been ingested, the antibody with the antigen, the antibody that has trapped the antigen has been ingested, and it forms a clathrin coated vesicle where the antibody is recycled and the antigen stripped off. This is early endosome, pH 6 to 6.5. As it is moving down the compartments, the pH is decreasing, and then these class 2 containing compartments fuse with these compartments. So we have this late endosome, pH is 4.5 to 5. Within this compartments, as you can see, when the class 2 containing MHC class 2 compartments containing the MHC class 2 molecule and the invariant chain moves to fuse with the late endosome, you can see digestion of the invariant chain is occurring, so that only a small fragment known as clip, which remains bound to the groove of the MHC class 2 molecule. Now what happens? This MHC class 2 fuses with the vesicle containing the peptide. There is a category of non-classical MHC molecule which is known as HLA-DM. This HLA-DM are present in the MHC class 2 containing compartments. It is with their help that the clip is removed and the antigen is loaded into the groove, and the class 2 molecules then present the antigen to the surface of the cells. So this was it. This is the synthesis of, this is occurring in the ER, okay? This is the synthesis of alpha and the beta MHC class 2 molecules. And once this alpha and beta class 2 chains have been formed, they associate, as I said, with the invariant chain. This invariant chain interacts with the class 2 peptide binding groove, preventing them from trapping or preventing them from being loaded with any self antigen that has gone wrong. Remember, for a antigen presenting cell, this is something you should remember. An antigen presenting cell has both class 1 and class 2 molecules. Keep this in mind. So in the endoplasmic reticulum, there are class 1 molecules also and there are class 2 molecules also. And if you remember, the TAP has an affinity for amino acids of 8 to 16 amino acid long. So when these amino acids enter the ER, if they are longer amino acids, they might have a tendency to bind to the groove of MHC class 2, which is also present in the ER of the APCs. To prevent this happening, to prevent the loading of intracellular antigen into the peptide binding groove of class 2 molecules, the invariant chain is present there so that no intracellular antigens can bind to the class 2 binding groove. In this form, it comes out of the ER in a vesicle, And then the invariant chain is gradually digested, as I said, within that MHC class 2 containing compartments, and only a small fragment known as clip remains. This MHC class 2 containing compartments also have another non-classical MHC molecule known as DM. This class 2 containing compartments, as we saw, fuses with the endosome, late endosome. After fusion, what happens? The HLA-DM removes the invariant chain for an antigenic peptide. So this exchange occurs, this is removed, and the peptide is loaded. And now we have a MHC class 2 molecule loaded with the peptide. So this is the endocytic pathway. So as you can see, we are having MHC class 2 invariant chain complex moving out into the through the Golgi, fusing with the endolysosome. During fusion, you can see the invariant chain has been digested. We have the MHC clip component. This is one part of the story. Meanwhile, in the other part, antigen was engulfed. As you can see, the antigen has been partially digested, and the endosome now fuses with the compartments containing these clip fragments, MHC clip fragments. Sorry, this is the MHC clip fragment. Very sorry. This is the MHC clip fragment. And as I said, in these compartments, HLA-DM is present. HLA-DM then removes the clip, loads the peptide, and you can see the peptide is loaded and presented. So this is in a nutshell the endocytic pathway. Just to keep it in mind once more, MHC restriction, exogenous pathway. That is, which is conducted by the antigen presenting cells in the exogenous pathway. When you're taking foreign pathogens, we saw that how the MHC class 2 molecules are processed, they mature, and how they're uploaded, how they're loaded with the peptide and uploaded on the surface. They always express their peptide to CD4 positive T cells. In the endogenous pathway or the intracellular pathway or the cytosolic pathway, the self-aberrant antigens or antigens of obtained from virus are presented to T cytotoxic cells. So these are the two basic differences between the MHC class 1 presentation and MHC class 2 presentation. Class 1 always presents to T cytotoxic cell. Class 2 always presents to helper T cells, CD4 positive cells. Now see, the output is also different. As you can understand, whenever T cytotoxic cell recognizes a peptide complex with MHC class 1, okay, so this will be MHC class 1, right? There's a mistake here. This will be MHC class 2. So whenever the CD8 positive T cell recognizes a peptide complex with MHC class 1 molecule, remember the function of the T cytotoxic cell is finally to kill this cell, because by then the immune system has understood that this cell contains a huge amount of aberrant proteins, and this cell itself needs to be destroyed. But whenever an antigen presenting cell presents its peptide via MHC class 2 to T helper cells, you can understand by the function that the antigen presenting cell is instructing the CD4 positive helper cell via signals to identify the antigen and to amplify the immune reaction. So it will lead to further immune activation. This will lead to killing of the target cell, but this will lead to further immune activation, depending on what kind of antigen is being presented to the CD4 positive helper cells. So that is a basic difference. And with this, we come to the end of our class. in nutshell, these are the two defined pathways. As you can see, the endogenous or the MHC class 1 pathway and the exogenous or the MHC class 2 pathway. Just to summarize, endogenous antigen is degraded by a constitutive proteasome or immunoproteasome, enters via the tap into the endoplasmic reticulum. The MHC class 1, in the endoplasmic reticulum, the class 1 molecules bind with Kynexin ERP57, then beta-2 microglobulin in an order. Kynexin dissociates, reticulin and tapasin binds. MHC captures the peptide and the chaperones dissociate, and you can see the MHC class 1 peptide is being transported from the rough endoplasmic reticulum to the Golgi complex and finally to the surface of the plasma membrane. When we are looking into the exogenous pathway, you can see we are having the MHC class 2 molecules, but the MHC class 2 molecules are in presence of invariant chain which binds to the class 2 alpha and beta chains, blocking the entry of any endogenous peptide into the binding groove. Then as you can see, the MHC class 2 containing the invariant chain, that compartment is routed through Golgi to the endocytic compartments. Meanwhile, the antigens have been engulfed. The antigens moved from acidic to acidic compartments. Fusion of this, during this movement of the MHC class 2 molecule towards these endocytic compartments, the digestion of the invariant chain occurs. Only a small fragment remains bound to the peptide binding groove, that is the clip. Fusion of this MHC class 2 containing the clip and the endocytic compartment occurs, and a non-classical HLA-DM, which is not shown here, molecule exchanges the clip for antigenic peptide, and MHC class 2 presentation is taking place. So whenever we have T helper activation, as you can see, there are two pictures shown here. This is the licensing of T helper cells. The dendritic cells are activating the T helper cells via MHC class 2 as antigen presentation. And this is again a dendritic cell activating a T cytotoxic cell by antigen presentation. So this was in a nutshell or summarizing the processing pathways, and with this we complete our function and structure and function of MHC class 1 and class 2 molecules.